Hello friends, welcome to ENTRANCEADVISER.com. Today's topic is Chapter 2 Reproduction in Flowering Plants. In this video, I will describe the anatomy and the pre-fertilization events of the plants. If you are new to my channel, press the bell icon and do subscribe to my channel. So let's start. Beginning with the overview, we know that reproduction is defined as a creation of a copy of something, or the act of sexual intercourse to create an offspring. The reproduction in flowering plants begins with the pollination, the transfer of the pollen from the anther to stigma of the same flower or to the stigma of the another flower on the same plant, which is known as the self-pollination, or from the anther of one plant to the stigma of another plant, which is known as cross pollination. Once the pollen grain lodges on the stigma, a pollen tube grows from the pollen grain to an ovule. Two sperm nuclei then passes through the pollen tube. One of them unites with the egg nucleus and produces a zygote. Other sperm nucleus unites with the two polar nuclei to produce an endosperm nucleus. The fertilized ovule develops a seed. As you can see in this picture, that beginning with the mitosis and then pollination, then fertilization, then a seed is developed. So we know that reproduction ensures continuity of the species generation after generation as the older individuals undergo senescence and die. The flowering plants also shows the sexual mode of reproduction and bears complex reproductive units as male and female reproductive units along with the accessory structures. The sexual reproduction is the type of reproduction that involves two different parents that belong to the opposite sex. This type of reproduction is seen in the plants also, as well as in animals, but in plants, sexual reproduction occurs in a more undeveloped manner, rather as not as that of the mammals, but it's more complex than the lower organisms. Before we learn more about the sexual reproduction in flowering plants, we must understand the anatomy of a flower. We know the flower is a fascinating organ of angiosperm. So, what is flower? Flower is a modified stem which functions as a reproductive organ and produces a ova or a pollen. A typical angiospermic flower consists of four whorls of floral appendages attached with a receptacle. The calyx, corolla, and rachim, the male reproductive organ consisting of stamens, and the gynoecium composed of ovary, style, and stigma. So let's discuss all this in much detail. Okay, we know the flower is a modified stem which functions as a reproductive organ and produces ova or the pollen. The structure of the flower and the flower is com- commonly borne and is short with long stalk called the pedicel. It has this is pedicel, and it has an upper solen region known as receptacle, that is thalamus. Okay, that is thalamus or storus. This part. The androecium and gynoecium are known as the contraceptive structures of a flower. The male contraceptive structure is the androecium. It comprises of the whorl and the stamens, as I said. And the stamen, it has a slim stalk known as a filament. As at its tip, it is contained a bilobed structure called the anther. Usually, these are bilobed anther contains two theca or dioecious isolated by longitudinal groove. The transverse section of any anther shows four-sided microsporangia, two in every flap. Microsporangia further form as pollen sac contained in the pollen sac or the pollen grains. We will be coming to this later on, but before that, what is calyx? It is the outermost whorl of the flower. It is composed of the leaf like green sepals, and the sepals are essentially green in color, but in some cases they are colored like the petals. Such a condition of calyx is called the petaloid. What is corolla? This is the second whorl of the flower and consists of a number of petals. The petals are generally brightly colored and sometimes the fragrant, which make the flower to become attractive. Now the androecium, it is the third whorl of the flower and is the male reproductive organ consisting of stamens. Each stamen is made up of filament and anther. The filament supports anther at its tip. This is this is and the gynoecium, this is the last and the fourth whorl of the flower and is the female reproductive organ of the flower. It occupies a central position on the receptacle and composed of ovary, style and stigma and the component parts called the carpels. This is the female reproductive organ, as you can see. Coming to the next, pre-fertilization, the structure and the events. Several structural and hormonal changes led to the formation and development of the floral primordium. Inflorescence is formed that bears floral buds and then the flower. In flowers, male androecium and female gynoecium differentiate and develops in which male and female gametes are produced. Stamen, microsporangium and the pollen grain. Stamen consists of a long and slender stalk called the filament and generally bilobed anthers. Each lobe contains of two theca dioecious, as I said. The anther is four-sided structure consists of four microsporangia, two in each lobes. Microsporangia develop further and become pollen sacs which contain the pollen grains. Microsporangium is generally surrounded by the four layered cells: the epidermis, the endothelium, middle layer, and the tapetum. The innermost tapetum nourishes the developing pollen grains. The sporogeous tissues, it is compactly arranged and homogeneous cells which are present at the center of the each microsporangium when the anther is young. Megasporogenesis, the process of the formation and the differentiation of microspores, pollen grains from microspore mother cells (MMC) by reductional division called the megasporogenesis. The cells of sporogenous tissues undergo mitotic division to form the mycospore tetrad. As the anther mature and dehydrate, the mycospore dissociate and develops into pollen grains. Now we know the anther is the male reproductive organ, or it is known as androecium, consisting of stamens, and it is the third whorl. Each stamen comprises of two parts, namely the anther, and this part is filament, the stalk. The tip of the anther is supported by the filament, and here the pollen grains are produced by the meiosis and disappear eventually. Anther is the four-lobed sac-like structure responsible for the pollen formation, and the filament, these are thread-like structure attached to the anther and they keep the anther in place. The transverse section of the anther in the microsporangia that further forms a pollen sac. The pollen sac contains the pollen grains. This is a diagram of a typical stamen and a three-dimensional cut section of an anther. Before that, you must know the functions of a flower. The flowers are the modification of shoot to perform the function of the sexual reproduction. The flowers of most of the angiosperms are shaped variously to help diverse mode of pollination. The flowers provide seed for germination of pollen, development of the pollen tube, formation of the gametes, and fertilization. The ovary part of the carpel gets transformed into the fruit, and the ovules are transformed into the seeds after fertilization. Some floral parts, like the calyx and the various modification in ovaries, help in the dispersal of the fruits and seeds. This is an enlarged view of one mycosporangium showing the all layers. A matured dissected anther is described here. You can see the pollen grains, and this layer is epidermis. This layer is endothecium. These are the middle layers. This is the microspore or mother cells, and these are the inner cells tapetum which nourishes the cells. We know the pollen grains or the dust grains are round in shape. It consists of two layers, external exine and internal intine. The intine is thin and continuous layers made up of cellulose and pectin. The exine is composed of the natural compound called as sporopollenin, and it is hard and resistant natural compound. No one protein and is known as the base of this protein, and the exine consists of a typical pore called the germ pore. As the dust grain develops, it consists of two cells and a greater vegetative cell and a minute generative cell. And the generative cell isolates mitotically into two male gametes. So, as we know, the tapetum, the cells are the multinucleate, and this layer is one of the innermost layers of the cell wall. We know the middle layer is composed of, this is necessary to know that the middle layers, it is composed of 3 or 4 layers of the thin wall cells. These are situated just below the endothelium. And the endothecium, and this layer is the inner to the epidermis. Some cells develop on the end of the endothelium, and the epidermis is the protective outermost single layer. The cells of the epidermis is arthroperium developed of fibrous thickening, and the epidermis is designed as an exothecium. And the formation of pollen grains, this will be much easier to know as the diagram will be more clear. The young anther soon becomes a four-lobed as it comprises of a homogeneous mass or the parenchymatous cell surrounded by the epidermis. Each archaeosporial initial present in the hypodermal cells divide into the inner primary sporongenous cell and the outer primary parietal cell. Each primary sporogenous cells divide to produce microsporocytes. Each microspores mother cell divides the meiotically to form pollen grains. As you can see, this is the mother cells, and each microspore mother cells divides meiotically to form pollen grains. What is microgametosis? The development of the male gametophyte. Microspore is partially developed at this stage, and it is the first cell of the male gametophyte. The cell wall of the pollen grain is made up of exine, the outer layer, and the intine, the inner layer. The outer layer is made up of sporopollenin, and the inner layer is made up of pectose and cellulose. Okay, hope you understood all this. Now coming to the pre-pollination development. The microspore starts germinating in situ. A smaller generative cell lies next a lying spore wall. A callus layer is placed around the generative cell, and the callus cell layer then dissolves. Then the pollen grains are shed from the anther. The liberated pollen grains are transferred to the stigma through pollination. The pollen grain present on the stigma absorbs water and swells within a few minutes. The wall of the pollen tube secretes hydrolytic enzymes and exogenous pectinases to create a passage for its entry. The generative and vegetative are carried by the pollen tube. The generative cell divides to form non-motile male gametes. Tube's nucleus may disintegrate as there is no important function. The pollen grains represent the male gametophytes. We know the pollen grains are made up of two cell layers. One is exine and one is intine. You can see on this picture that this is exine and the inner layer is intine. This is germ pore, and this is generative cell. And this is vegetative nucleus, and this is generative nucleus. The thickening of nucleus is taking place and before within the intine. So coming to a structure of a mature pollen grain. A mature pollen grain consists of two cells with the nucleus, vegetative and generative. The vegetative cell is bigger, abundant food reserve, large irregular nucleus, and the responsible for the development of a pollen grain. And the generative cell is small, involves in syngamy fusion with an egg and dense cytoplasm and the nucleus. In this picture, you can see clearly with the haploid nucleus, it is a young pollen grain, and then the mitosis occurs, then the generative nucleus divides, and tube nucleus also forms, and a mature pollen grain occurs. And this is a detailed diagram, schematic diagram showing that the pollen mother tube, the diploid, then mitosis 1 occurs, then meiosis 1 occurs, then meiosis 2 occurs, the secretion of walls occurs, and then a mature pollen grain develops. So, pollen grains are of many species. Example, Parthenium causes severe allergies and the bronchial diseases in some plants and lead to chronic respiratory disorders, asthma and bronchitis, etc. The pollen grains are rich in nucleus and are used as a pollen tablets as food supplements. The viability of pollen grains varies within the species to species and should land on stigma before this period to germinate. Pollen grains of a large number of species are stored in liquid nitrogen at a temperature of minus 196 degrees Celsius called the pollen bank. So, what are germ pores? The apertures on the exine within the where the sporopollenin is absent. It forms a pollen tube, and the plasma membrane surrounds the cytoplasm of the pollen grain. Now coming to the gynoecium. Gynoecium, it is a female reproductive organ and the last whorl of the flower. It composed of the pistil and occupies the central position of the thalamus. The stigma, style, and the ovary are the components of the pistil. The ovary produces ovules internally through meiosis. Ovules produce megaspores which in turn develops into female gametophytes. As a result, the egg cells are produced. The gynoecium the consists of single pistil known as monocarpellary and the more than one pistil known as polycarpellary, which may be fused syncarpous or free apocarpous. The multicarpellary or syncarpous pistil are papaver and multicarpellary and apocarpous pistil is mycelium. Each pistil has a three parts stigma, style, and ovary. From this picture, it is clear it consists of a three parts stigma, style, and ovary. Inside the ovary, the ovarian cavity is present. Okay, locule. It is known as locule. The placenta is located inside the ovarian cavity. This is the placenta and this is ovule. And the megasporangia or ovules arise from the placenta. You may also refer to this diagram also, which is a more scientific or more schematic than this, and for the carpels you may see the simple aggregate and multiple carpels. There is the monocarpellary or polycarpellary. Monocarpellary means the gynoecium consists of a single pistil, for example peas and beans. Multicarpellary, here the gynoecium comprises more than one pistil. And the syncarpous, as I said, it is gynoecium with combined pistil, for example the tomato and cucumber. And apocarpous pistils are it is the this the gynoecium with the free pistil, example the lotus venica. If we see in detail, the pistil consists of three parts. The ovary is the chamber where the ovules or eggs are stored waiting for the fertilization. And the stigma is attached to the top of the carpel where the pollen from the other flowers lands. Here the pollen lands. And this is the style, and it is a tubular structure you can see that connects the ovary and the stigma, and it is responsible for the transportation of the pollen from the stigma to the ovary and holding the stigma in the place. At last, the megasporangium or the ovule. What is megasporangia? The ovule is connected to the placenta with a stalk called the funicle. It yields the megasporocytes that form the megaspores. The megasporogenesis is the process of arrangement of megaspores from the megaspore mother cell. The megaspore mother cell divides meiotically. Uh, One of the three megaspores is utilized while the other three degenerate. The function of the megaspore forms from the female gametophyte. The ovule is a small structure attached to the placenta, as we described earlier. The funicle is the stalk by which the ovule is attached to the placenta. Hilum is a junction between the ovule and the funicle. Integuments are the protective envelopes and micropyle, small opening at the tip of the ovule into where the pollen tube enters. Chalaza is the basal part of the ovule. Nucleus 2n, the mass of the cells enclosed in the integuments, has the abundant food for reserve. From this diagram, you can see clearly that the megasporangium and uh, it is a first of all this is a flower. Then if the microsporangium or the anther develops, then it forms the pollination. And if you see in this way, the gynoecium develops and the immature ovule, the megasporangium also develops and it fuses. And after that, the embryo, after fertilization, the embryo sac and then the seed, then young sporophyte develops. And this is a diagrammatic view of a typical anthroporous ovule. And this is the hilum, this is the funicle, and this is the micropyle. And this is micropylar pole, outer integument, inner integument, nucleus, and embryo sac at last. This is the basal part or chalazal pole. And and this is a more simplified view and an overall diagram to remember clearly the pollen grains. In this diagram, you can see the pollen grains enters from here and then drops down here, and this is a pollen tube, this is pollen receptor, those are antipodal cells, and this is ovule, this is a generative cell, this is a synergids. I'm coming to these terms in a moment. Wait, just and pistil and the polar nuclei. So, what is megasporogenesis? The process of formation of megaspore from the megaspore mother cell by the meiotic division is known as Megasporogenesis. The process takes place in the ovule. The ovule differentiates a single megaspore mother cell MMC in the micropylar regions of the nucleus. MMC undergoes meiotic division that results in the production of the four megaspores. In the most of the flowering plants, three megaspores degenerate. One megaspore develops into female gametophyte embryo sac. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles to form the nucleate embryo sac. Two more sequential mitotic division results in two nucleate embryo sac. And six of the eight nuclei surrounded by the cell wall and the remaining two nuclei are called polar nuclei or situated below the egg apparatus. These cells are grouped as the micropylar and to constitute egg apparatus, and three cells are chalazal and forms the antipodal cells at maturity, and the embryo sac is 8 nucleate and 7 nucleate. These are the schematic diagrams showing the megasporogenesis in an overviewing manner, and this is the ovary before the megasporogenesis. After that, meiosis takes place, then mitosis. Okay, then polar nuclei is generated. This is the ovary after megaspore, and this is a matured one. Before maturation, there was no differentiation of such like this. You can see. This is also a diagram showing the development of an ovule. From outside, it will look like this, and if we cross section it, it will look like this. These cells developed are called synergid. These are egg, and these are antipodal cells, and these are polar nuclei. This book, this you may find also this diagram in your book, and these are the parts of the ovule showing the large megaspore of the mother cells are dyad and the tetrad megaspores, the A diagram. The B diagram is showing the 2, 4, and 8 nucleate stages of the embryo sacs and the mature embryo sac and the mature embryo sac, and the diagrammatic view of a representation of a mature embryo sac in a large view as we have seen in the earlier slide. This is a chalazal end. These are antipodals and these are polar nuclei. This is central nuclei, central cell. This is egg. These are synergids. This is filiform apparatus at the micropylar end. So, thank you for listening. See you in the next video where I am going to describe about the pollination. So, take care and bye bye.